Chapter 12, Section 3, Translation. The sequence of nucleotide bases in an mRNA molecule serves as instructions for the order in which amino acids should be joined together to produce a polypeptide. However, anyone who has tried to assemble a complex toy knows that instructions generally don't do the job themselves. They need something to read them and put them to use. In the cell, that something is a tiny factory called a ribosome. The decoding of an mRNA message into a polypeptide chain is known as translation. Translation takes place on ribosomes. During translation, the cell uses information from the messenger RNA to produce proteins. Refer to Figure 12-18 as you read about translation. Part A. Before translation occurs, messenger RNA is transcribed from DNA in the nucleus and is released into the cytoplasm. Part B. Translation begins when an mRNA molecule in the cytoplasm attaches to a ribosome. As each codon of the mRNA molecule moves through the ribosome, the proper amino acid is brought into the ribosome by tRNA. In the ribosome, the amino acids is transferred to the growing polypeptide chain. Each tRNA molecule carries only one kind of amino acid. For example, some tRNA molecules carry methionine, others carry arginine, and still others carry serine. In addition to an amino acid, each tRNA molecule has 3 unpaired bases. These bases, called the anticodon, are complementary to one of the mRNA codons. In the case of the tRNA molecule for methionine, the anticodon bases are UAC, which pair with the methionine codon, AUG. The ribosome has a second binding site for a tRNA molecule for the next codon. If that next codon is UUC, a tRNA molecule with an AAG anticodon would fit against the mRNA molecule held in the ribosome. The second tRNA molecule would bring the amino acid phenylalanine into the ribosome. Part C. Like an assembly line worker who attaches one part to another, the ribosome forms a peptide bond between the first and second amino acids, methionine and phenylalanine. At the same time, the ribosome breaks the bond that had held the first tRNA molecule to its amino acid and releases the tRNA molecule. The ribosome then moves to the third codon, where a tRNA molecule brings it the amino acid specified by the third codon. Part D. The polypeptide chain continues to grow until the ribosome reaches a stop codon on the mRNA molecule. When the ribosome reaches a stop codon, it releases the newly formed polypeptide and the mRNA molecule, completing the process of translation. Now refer to Figure 12-18, Translation. During translation, or protein synthesis, the cell uses information from the messenger RNA to produce proteins. The cell uses all 3 main forms of RNA during this process. Remember, Part A, messenger RNA. Messenger RNA is transcribed in the nucleus and then enters the cytoplasm and attaches to the ribosome. Part B, Transfer RNA. Translation begins at AUG, the start codon. Each transfer RNA has an anticodon whose bases are complementary to a codon on the mRNA strand. The ribosome positions the start codon to attract its anticodon, which is part of the tRNA that binds methionine. The ribosome also binds the next codon and its anticodon. Part C, the polypeptide assembly line. The ribosomes join the two amino acids methionine and phenylalanine and breaks the bond between methionine and its tRNA. The tRNA floats away from the ribosome, allowing the ribosome to bind to another tRNA. The ribosome moves along the mRNA, binding new tRNA molecules and amino acids. Part D, completing the polypeptide. The process continues until the ribosome reaches one of the 3 stop codons. The result is a complete polypeptide.